Hello. I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get accepted into medical school and other professional programs. And welcome back to another episode of MCAT Bites. Today, we're talking about energy production and ATP. Understanding how our cells generate and utilize energy is fundamental to grasping many, if not all, physiological processes, and will be crucial for your MCAT success on the biochem and chem phys sections. Heck, there are probably even some questions on psych-soc that deal with this. Because of the importance of this, I would honestly recommend watching this video twice. Once before you dive into the details of glycolysis, beta oxidation, and the pentose phosphate pathway, and then once after you've learned all that. So the theory behind this is I want to give you a broad overview, then you learn the details, and then you can connect those details to broader physiological functions. By the end of this video, you should have a solid understanding of ATP production, its role in cellular activities, and some other key pathways involved from a bird's eye view. So let's dive on in. Start with what is ATP? ATP, or adenosine triphosphate, is often referred to as the energy currency of the cell. It consists of adenine, a ribose sugar, and three phosphate groups, 1, 2, 3. The bonds between these phosphate groups store a significant amount of energy, which can be released to fuel various cellular processes. ATP is essential for muscle contraction, active transport across cell membranes, and almost all of the important biochemical reactions in your body. So how do we make this important thing? A lot of different ways. ATP is produced primarily through cellular respiration in us, which includes glycolysis, the citric acid cycle, and oxidative phosphorylation. These processes convert glucose and other nutrients into ATP, providing energy needed for cellular functions. So we take the glucose in the cytoplasm, we'll make some pyruvate. Don't worry about the exact steps to do this. There's 10 of them. We'll go into that later. Once we have the pyruvate, that can cross into the mitochondria, where it can enter the citric acid cycle, also known as the Krebs cycle. The Krebs cycle is going to make some ATP directly, but also a thing called NADH and FADH2. This can go somewhere else inside of the mitochondria called the electron transport chain, where the NADH and FADH2 can be used to produce ATP. And it does this through a hydrogen pumping gradient, which again, we will also cover in more detail shortly. I don't want to bog you down in details quite yet. So the first step of glycolysis occurs in the cytoplasm, like we mentioned, and involves the breakdown of one glucose molecule into two molecules of pyruvate. And let's see why that makes sense. Well, pyruvate has 3 carbons, such as these circles are representing. So if we start with 6 carbons of glucose, cut that in half by 2, okay, we have 2 sets of 3. So that's why we have 2 pyruvates. This process generates a net gain of 2 ATP molecules and 2 NADH molecules, which can be used for later stages of cellular respiration in the mitochondria. What's nice about glycolysis is it does not require oxygen, making an anaerobic process. Next up, we have the citric acid cycle, also known as the Krebs cycle. This will take place in the mitochondria. Here, each acetyl-CoA molecule produced from pyruvate enters the cycle from the top here and is oxidized, producing 3 NADH and 1 FADH2, and 1 ATP per cycle. And remember that it'll cycle twice because we produced 2 pyruvate and therefore 2 acetyl-CoA. And these high energy electron carriers, NADH and FADH2, are crucial for the next stage of ATP production, which happens in, drumroll please, the powerhouse of the cell, as did the citric acid cycle though. This is where oxidative phosphorylation occurs, inside the inner mitochondrial membrane, and it consists of the electron transport chain, or ETC, and chemiosmosis. It's a fancy way to say pumping hydrogen across the gradient. NADH and FADH2 are going to donate their electrons to the electron transport chain, which will then create a proton gradient by literally pumping protons across the membrane. And then the flow of protons back through ATP synthase is actually going to be providing the energy needed to stick a phosphate onto a DP to create a TP. All in all total, this process is going to generate the most ATP, yielding approximately 34 ATP molecules per a single glucose. That's pretty good. That's about a 33% perfect energy conversion. So we've made ATP, what the heck are we going to do with it? Well, we can use it in a bunch of different functions, including muscle contraction, active transport, and biosynthesis. In muscle cells, ATP binds to myosin, allowing it to interact with actin filaments and produce muscle contractions. In active transport, ATP provides energy to pump ions and molecules against the concentration gradients. So it's essential for maintaining cellular homeostasis. And that's all for new content in this video. Now let's recap this by doing some practice problems. Starting with question 1. How many ATP molecules are produced from one complete oxidation? How many ATP molecules are produced from the complete oxidation of one glucose molecule during cellular respiration? Take a few moments to think about this. I'll give you the answer in 3, 2, 1. Boom. And here's where things are kind of complicated because people are always reporting a range, anywhere from 32 to 38 ATP molecules, depending on how you want to calculate it. And depending on how your biochemistry professor taught it, you'll get a number in that range. But don't worry, the MCAT accounts for that and you will be okay. In this example, we're producing 2 ATP net during glycolysis, but a total of 4 ATP. We just wrote 2 for net, but I asked produced, not net, so we'll say 4. Then we produce another 2 ATP down here in the Krebs cycle, and then we produce 28 ATP while in cellular respiration. We add that up, we get 34 ATP total. Now let's move on to problem 2. I want you to talk to a friend, talk to yourself, write it down, but describe the role of ATP in muscle contraction. Take a few moments, and when you're ready, I'll give you the answer in 3, 2, 1. Here it is. And this is a great time to pause the video and make some screenshots to make new Anki cards. The MCAT loves asking about power strokes. So essentially what's going on here is ATP will bind to myosin, which allows it to detach after a power stroke. ATP is then hydrolyzed to ADP in an inorganic phosphate, providing the energy for myosin to return to its high energy state and reattach to actin for the next contraction cycle. Now, finally, how does ATP facilitate active transport in cells? I'll give you the answer in 3, 2, 1. ATP provides the energy needed for transport proteins to move ions and molecules against their concentration gradients. That's the key word or phrase, against the concentration gradient. For example, the sodium-potassium pump uses ATP to transport sodium out of the cell and potassium into the cell, maintaining essential electrochemical gradients. And we know that there's more potassium inside the cell and sodium outside the cell. So this takes energy because it's not something that'll just naturally happen. We're fighting entropy here. And whenever we're fighting entropy, it's going to cost a little bit of ATP. Understanding energy production in ATP is crucial for mastering cellular biology and metabolism for the MCAT. ATP serves as the primary energy currency of the cell, fueling various essential functions that we've talked about. The intricate processes of glycolysis, the citric acid cycle, and oxidative phosphorylation ensure that our cells have a constant supply of this vital molecule. Keep studying these pathways and you'll be well prepared for the MCAT and your future medical career. Again, I do recommend watching this video twice, once at the beginning of your studies, and then again after you've mastered glycolysis, beta oxidation, and the pentose phosphate pathway. But thank you so much for watching our video on energy production and ATP, and I will see you next time.